Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Khadijah binti Abu Sa'ama, and today I'll be presenting on isolation and identification of soil actinobacteria with antimicrobial activity from Kuala Selangor Nature Park by using heat treatment. What is actinobacteria? Actinobacteria are high TC content bacteria that produce hyphae and spores similar to fungi. They play a crucial role in soil health by producing antibiotics and other important compounds. These bacteria are found in diverse environments such as soil, water, and extreme conditions. This study focuses on the mangrove soil of Kuala Selangor Nature Park, a biodiverse area with various habitats and ecological functions. So now, what is antimicrobial resistance is? AMR is a global constant where bacteria evolve to resist antibiotics, making treatments less effective and increasing mortality rates. This happens when bacteria break down antibiotics, block their entry into cell, or change their targets. Infections like MRSA and also Neisseria gonorrhea become harder to treat, leading to fewer treatment options, higher healthcare costs, and also longer hospital stays. There's an urgent need for new antibiotics and better strategies to monitor and control resistance. Addressing the significant global health threat posed by antimicrobial resistance, pathogens are increasingly resistant to treatment options, including drugs against bacteria, viruses, fungi, and also parasites. Actinobacteria, crucial for producing antibiotics, present challenges in isolation due to their slow growth and competitive nature in soil. Therefore, methods like heat treatment are essential to enhance their study and also unlock their antibiotics. In our study, I have formulated the following research question. The first one is, what are the specific type of bacteria found in the mangrove soil of Kuala Selangor Nature Park with a focus on actinobacteria? And second one is, how does heat treatment affect the abundance and diversity of actinobacteria in soil samples collected from Kuala Selangor Nature Park? And the last one is, do our isolate obtained from Kuala Selangor Nature Park able to produce antimicrobial properties against common pests? It has the following objective. The first one is to identify and classify the specific type of bacteria present in the mangrove soil of Kuala Selangor Nature Park with a particular emphasis on actinobacteria. The second one is to evaluate the impact of heat treatment on the abundance and diversity of actinobacteria in soil sample collected from Kuala Selangor Nature Park. And the last one is to assess the antimicrobial properties of actinobacteria isolates obtained from Kuala Selangor Nature Park against common pathogens. To address our research question, we employ the following methods. Firstly, sample will be collected from mangrove soil at Kuala Selangor Nature Park. Then it will be divided into two portions, with and without heat treatment. Both of it will be air dried for 1 week. Samples that undergo the heat treatment will be heated for 30 minutes at 65 degrees Celsius. Next, in order to isolation of actinobacteria, serial dilution of the soil sample were prepared on plated on starch casein agar, which is SCA. The plates were incubated at 27 degrees Celsius for 1 to 4 weeks to allow for actinobacteria growth. After that, we will proceed to screening the antimicrobial activity. The first one is primary and secondary screening. Primary screening is to identify isolates with antimicrobial activity. Primary screening performed on MHA agar using perpendicular streak method. Isolates showing activity in primary screening undergo secondary screening to produce crude extract, which are tested against pathogen to confirm their antimicrobial properties. Crude extract tested by using agar well diffusion method. The next one is the MIC test follows secondary screening using the crude extract to determine the minimum concentration needed to inhibit the growth of test bacteria. This step quantified the antimicrobial potency observed in the screening phases. MBC testing builds on the MIC result by identifying the minimum concentration required to kill the bacteria, providing a complete picture of the antimicrobial efficacy of the extract. For molecular identification, we extracted DNA, performed jet electrophoresis, determined DNA purity and concentration, amplified the 16-stranded RNA gene using PCR, clean up the PCR product, and sequenced the 16S rRNA gene. This study is important because it identifies and classifies bacteria, especially actinobacteria, in the mangrove soil of Kuala Selangor Nature Park. This enhances our understanding of microbial diversity and could lead to discovering new useful compounds for drug development. This also helps protect mangrove habitats and address the urgent need for new antibiotics to address antimicrobial resistance by exploring the antimicrobial properties of actinobacteria. The finding could lead to new antibiotics and improve soil management practices. Through this research, we expect to identify and classify specific type of bacteria in the mangrove soil of Kuala Selangor Nature Park with a focus on actinobacteria, enhancing our understanding of microbial diversity. Second, determine how heat treatment affects the abundance and diversity of actinobacteria in soil samples, optimizing conditions for their isolation and also study. And the last one is, we expect that to discover actinobacteria isolate with antimicrobial properties against common pathogen, potentially leading to the development of new antibiotics and improved soil management practices. So this is my Gantt chart. So that is all from me. Thank you.